Hola, my name is Tveit and I'm a postdoc at the Norwegian Research Center in the Energy Department at the Computational Geoscience and Modeling Group. In this presentation, I will talk about ad-micp, an MRST model to study microbially induced calcite precipitation with the application for CO2 leakage remediation. I have divided the presentation in the following parts. So first, I will mention briefly the conceptual model for this technology. This presentation will be questions-free. For that, you can refer to the publications. After, I will show the content inside the GitHub repository. We'll spend most of the time running two examples, one in GNU Octave and the other one in MATLAB. Then, I will say a few words about ad-micp and the Open Porous Media initiative. Finally, I will mention the concluding remarks and more details about the ongoing work in this project. Microbially induced calcite precipitation can be defined as a biochemical process that induces calcium carbonate precipitation within the soil matrix. Then, the application we're interested in this project is to use this process in order to remediate leakage paths for CO2 storage. For the micp conceptual model, we consider a constant temperature reservoir saturated with water, where calcite and biofilm only occur on the rock walls. That is, in the space domain, there are one liquid phase, water, and three solid phases, the biofilm, calcite, and the rock matrix. The microbial medium, growth components, and cementation solution are dissolved in water prior to injection, and they are transported only in the water phase by advection and dispersion. The biofilm and calcite are assumed to be impermeable and incompressible. The governing processes in the biofilm are growth, death, attachment, and detachment. We consider the limiting factor in the growth solution to be the oxygen. This assumption can be justified since oxygen has a limited solubility in water, while the other components can be injected at higher concentrations. Assuming enough calcium concentration in the water, we model the calcite formation as a function only dependent on the urea and the biofilm. This assumption can be justified since calcium can be injected together with urea in the cementation solution and will thus distribute in a similar manner, ensuring that both concentrations are high in the location where calcite precipitation is aimed. Then, in this simulation, the leakage paths are modeled as a porous medium with higher permeability, and we only consider scalar permeabilities. For details on the mathematical model, we refer to the publications. Okay, so here we're in the GitHub page of the ad-micp model. And here you can find, for example, the readme with all the necessary information about the previous changes of the other releases. Information about also, for example, the publications that you can relate to see the mathematical model. And in addition, you can see here this publication folder. In there you can find scripts in order to reproduce the figures that you might find on the publications. And the rest just follows the same structure as any MRST model when you have, for example, the models and so on. Okay, let us run the first example using Octave. Then we run the startup inside the MRST folder. After we go to the ad-micp model. Here I have added a new folder with two examples. So the first example, we're gonna run it here in Octave, and the second one using MATLAB. Then let us open this in the editor, and while we are seeing the content, let us run the simulation, make 1D. And now inside the editor, let's see. So for the required models, we're using the ad-blackoil, ad-core, and ad-micp. Like in other MRST examples, first we set up the grid. In this case, we'll have an injection well on the left and an outflow on the right hand side. Then we'll have a grid that is fine on the side when the micp processes are more relevant, and after it gets coarser towards the other end. Then we set up some rock properties and the fluid properties, and for the remaining model parameters we leave it inside the fluid structure. For example, the attachment rates, and so on. Here we also can give different porosity penalty relationships. The one we are using in this model is one such that a minimum value of permeability is reached when a minimum value of porosity, a critical value of porosity has been reached. Now, for the injection strategy, we use this M matrix. So, the first column, we give the time we're injecting each of the solutions. For example, for the microbial solution, will be 20 hours. After we get the time step, so we can have larger time steps when the wells are off. Then we give the injection rates. In this example, for simplicity, we give the same values, but one can play with different values. Then we have the concentration of the microbes, concentration of the oxygen, and concentration of the urea. So in this example, then first we inject the microbial solution, we close the well. After the oxygen growth solution, we close the well, and after the cementation solution, and we close the system. Then we add the injection well from the left-hand side, and in here, since we're using an MRST function to compute the velocities, that is called phase flux to cell velocity, then when we have the injection well on the boundary, we need to correct the values. That's why we give this the cell. Then we set up the outflow boundary condition on the right hand side of the domain. When we're setting the simulation schedule, then we use this M matrix that we defined before. And for example, for the total times number of time steps, this one we computed when dividing the time steps in each of the times we're injecting of the components. So the sum will give you the total number of time steps. Then we just set up the model and the solver. And here we can, for example, with this example, we're giving strict tolerances. Then we can just define the initial condition. In this case, there is not any of the components at the beginning inside the aquifer, but for example, if one can give a biofilm distribution initially inside the aquifer. And then we run the simulation. And here, since we're using Octave, then we're gonna use this MRST to VTK function in order to export the results. And we will use ParaView to do the visualization. So now the simulation has been complete. And then we open the ParaView. And in ParaView, then we open the output files. So we go to Documents, MRST, Models, ad-micp, Symposium, VTK. So we open this states PVD. And here we have import the data. For example, if we want to see the microbial dynamical evolution, we select the max, and after we put it to run. And similar to MATLAB, we can, for example, plot the 1D profiles. So, for example, for the microbes, we have here, we put it to play, and we can visualize another output for at the same time. For example, for the oxygen, that's the next one to be injected, and so on. So now, this MRST to VTK function can be also used with other MRST models. And here I have run some simulation before using the geothermal model, and we can see how this works. So we go to Documents, MRST, Models, we go to the geothermal examples, and I run the geothermal example state, and after I apply that function in order to export the VTK format. And here you see we import the results, and we can do the visualization. For example, for the temperature, we can go to the final profile, and this ParaView is very flexible, that you can change, for example, the color bars to have a nicer one, and also you can do the visualization of the wireframe of the grid, and so on. So then this example was planned in order to show you how we can run this ad-micp in Octave using this MRST to VTK function to export the results in ParaView, and using ParaView to do the visualization of the results. Okay, for the second example, let us use MATLAB. So we run the startup inside the MRST folder. We go to the ad-micp. And just to remind, the first example we run it in Octave, and we produce these VTK files. Now let us open this Mix3D. So in this example, we're going to set up a 3D system in order to assess the CO2 leakage before and after the MIC treatment. For doing this, we're going to use this mesh. If you haven't got this mesh before, you can just uncomment these lines and get it. To do the assessment of CO2 before and after treatment, we're going to use one of the existing models already implemented in MRST in the CO2 lab model. And since we're using MATLAB, we can use the GUI to do the visualization of the results. So first, we're gonna set up the reservoir geometry. And I'm not gonna go into detail because of the time, but basically we'll have one injection well, one leakage path, and an upper and lower aquifer, and cap rock in between. And when we do the grid, we're gonna remove the cells when there is cap rock. So if we run this, here is this mesh where we will have the leakage path somewhere located in this region, and the injection well on the left, and open boundaries on the left and right hand side. So here the grid has been done, and then we have set up again the rock and fluid properties and the model properties inside the fluid structure. To do the CO2 assessment, as I mentioned, we're going to use one of the existing models in MRST. This is basically a copy-paste from one of those examples, the basic 3D example. And here we just change the injection rate and give a shorter schedule in order to run the simulation fast. So if we run this, as we can see here, we have the injection of the CO2 into the aquifer, and we have here the leakage path, and here on the gap is cap rock. We have removed the cells, and now the simulation is done. Here we are computing the leakage rate in the leakage path after the simulation has been complete. And the idea is because we want to compare after the MIC treatment how much that leakage has been reduced. Now let us run the cell for the MIC treatment. In order to see the two figures at the same time, we can just go here, view, left, right. Okay, so here first we start with injection of the microbial solution, and that's gonna lead to some biofilm formation. While the simulation is running, we can also just explain here. As in the 1D example, we give the injection strategy here. When in the first column we have the time when injecting each of the solutions. When the wells are off, we can have larger time steps. After we have the injection rates and the microbial concentration, oxygen concentration, and the urea concentration. So here now, as we see, the gross solution start to be injected, and with this we want to stimulate the formation of the biofilm on close to the leakage path. Here we notice we define two wells. It's gonna be the upper and lower part of the well, and this is because previous studies have shown that if this technology would like to be applied on the field, it seems that the best way to do it is to inject all the components or solutions on the upper part of the well, and on the rest part just water in order to transport the components, and this is in order to avoid the whole encapsulation of the well due to the MIC process. Okay, so now in the simulation, the cementation solution starts to be injected, and that's going to lead to some formation of the calcite and hence permeability reduction, and as you can see. Like in the 1D example, then we use the M matrix to set up the schedule. And here we have the upper and the lower part of the wells. We need to give the correct fraction of the injection rates. And now we have this function getPlot after step mic, and this one produces these two figures, and these two numbers is just gonna change the view of these plots. And once the simulation is done, then we can do the CO2 assessment. For that, we can compute the the new porosity and hence the reduced permeability, and run the CO2 simulation again. And when the simulation is done, then we can compute the reduction of the leakage. And finally, we can use this plot toolbar to do some inspection of the results, and we can also plot then the leakage before and after treatment in order to compare. Now the simulation is complete. Then we can run this CO2 assessment again. So again, here we're injecting, we have open boundaries and the leakage path, and now the simulation is done. Then we can compare the leakage before and after treatment. Here in the red we have the leakage before the treatment, and in orange we have the leakage after the treatment. As we can see here, using just this one phase, that is injection of the gross solution, macro solution, gross solution, and cementation solution, it leads to some reduction of the leakage, but not too much significant. So then one can just give extra treatment phases. For that we refer to the paper, so you can see one way to do it. And the other figure that we get is this here, and here, for example, you can just check the final calcite formation as in this example. Then we're aiming to have the penalty reduction in the leakage path. About the compatibility of ad-micp with GNU Octave in different operative systems, if you have Octave 6.3 either in Windows or Linux, it should work. I have tested using Ubuntu and Windows 10. If you have Mac OS, I would recommend to use Octave 6.1 because the 6.3 has some issues if you want to use this mesh, but 6.1 it works with this mesh. In general, if you want to use Octave and MRST, I would recommend for the visualization of the results to use the MRST to VTK function in order to use ParaView, and for making grids to use this mesh. Inside the publications folder, you will find the folders paper A and paper B. In paper A, you will find all these scripts like 1D, 2D, and so on, in order to run the examples and produce the figures in the paper. For the visual art, what I used was IPE, a free drawing editor for creating figures in formats like EPS, and one can use LaTeX commands for the text. The next step in this project was to implement this MIC mathematical model in OPM, the Open Porous Media Initiative. For that, we have this paper B. Once we have done the implementation, the natural step is to compare the simulation results in MRST and OPM. In here, we present the simulation results in a 1D domain. The blue dots are the results you get by simulating using OPM, and the solid lines with MRST. As we can see, there is a good agreement between both implementations. Then, in the folder paper B, you can find the PVD files produced by simulating using OPM and the scripts to run the simulation using MRST and produce all these figures. So now, what is the current work in this project? One of the main objectives of this project was to implement a MIC mathematical model in OPM. For this, first we implemented the model in MRST for prototyping, but the approach we are following now is to use MRST to build the grids and exporting the grids in a GRDECL file to be read in OPM. So then, in OPM, we run the simulations. For example, here, we have a similar sequence as in the 3D example we run in MATLAB, where the CO2 leakage is assessed before and after MIC treatment. For visualization of the results, we are using Python. Then, the main idea is to have Python scripts to set up the grid using MRST, run the simulations using OPM, and make the figures using Python. Just to conclude then, we have presented here an MRST model to study microbially induced calcite precipitation at the field scale. This model is fully compatible with GNU Octave, and as we saw, it includes several examples from the publication. As part of the current work is to have the Python scripts to produce the grids using MRST and run simulations using OPM. The aim is to make the code available in the coming OPM release in November and using the implementation to run some optimization studies. So for the last slide, here we can see all publications produced in this makeup project, and here are the references for paper A and B in the ad-micp model. For this current work and the Python scripts, the idea is to make them available in this GitHub repository. And with this, I would like to thank you for your attention, and I'm ready to answer any questions you might have.